Hi, my name is Samir Patel. Hi, the interaction I'm going to be discussing is the interaction between Escherichia coli, or E. coli for short, and human intestinal cells. E. coli has a lot of different types. Mostly, we're only going to be talking about two different types. Enteropathogenic E. coli, or EPEC for short, and that's usually causes infantile diarrhea. It's really common cause for it. The other subtype is enterohemorrhagic E. coli, EHEC for short, and that causes hemorrhagic colitis and hemolytic uremic syndrome in adults. So exactly how does it cause diarrhea? Both of these are just fancy diarrhea. One just is bloody diarrhea, and the other one's a more watery diarrhea, the infantile one, enteropathogenic one. The way it does cause it is it leads to pedestal formations in the surface cells of our small intestines. So exactly how does it cause the surface to change and produce these pedestals is what we're going to be discussing. And it usually does that by rearranging the actin. So let me talk a little bit more about what actin is. Actin and the proteins that interact with it form an indispensable network in the cells. Actin is a part of the cell cytoskeleton. This dynamic filament is crucial for a number of cellular functions, including locomotion, cell division, shape changes in cells, which is what we're going to be talking about, and various forms of intracellular motility. So control of the site-specific assembly and disassembly of the actin filaments is critical for directing these activities to the appropriate position in cells. So I drew a picture here of just prokaryotic versus eukaryotic cells. The cell on the left is the prokaryotic cell, which is E. coli, and the one on the right is just a eukaryotic cell. You can see that the eukaryotic cell is much bigger, 10 to 100 micrometers in length, and the prokaryotic cell of E. coli is much smaller, 0.1 to 10 micrometers. Since this project really isn't about the differences between these two cells, let's just get straight into the interaction between these two. So the interaction actually happens here, if you see my cursor. It happens between the two surfaces of the two cells. So the way that these pedestals form, it takes multiple steps. And first we're going to talk about EPEC. EHEC does it a little bit differently. Well, I'll get to that at the end. EPEC, what it does is it first binds. It comes along and it binds to this cell through bundle-forming pili. And once it's binded, it connects to the cell via a type 3 secretion system. Once that's there, it secretes a TIR protein, also known as the translocated intimin receptor protein. That TIR protein, which is made in the bacteria, will come along, it'll come into the cell, and what it does is it puts itself into the plasma membrane of the surface intestinal cells. And it acts as a receptor for intimin, which is a surface protein on the bacteria. Once this intimin binds to the TIR, it's what starts the whole cascade of events. Once it binds, the very first thing that'll happen is our own bodies, our own cells, tyrosine kinases will come and phosphorylate the intracellular portion of the tear. And once that gets phosphorylated, kinases, that's what they do. They phosphorylate. It's just a fancy word for attach a phosphate on. Anytime you see the word kinase, that's what they're doing. So it just basically attaches a phosphate here, which kind of activates it. And then it'll recruit a GTPase to polymerize the actin. The name of the actual GTPase and the actual protein here is NWASP. That might be a little bit too in-depth, but I just wanted to throw that out there. That's actually what it's called. And that will promote the actin to polymerize and form little chains here. If you see these 4 chains here, they're all different little actin filaments just bound together. And this will actually, once this happens, they usually do like 4 to 5 chains, sometimes 3 chains. But once they actually are formed, it will create this uprising in the cell at the top, if you see here. And that is actually what the pedestal is. So first you got to get these chains. Once those chains come, it'll create an upward rise, an outpouching of sorts. And that's usually anywhere up to about 10 micrometers in length. That's actually what creates the diarrhea. That'll lead to effacement of the small intestines. Effacement is just a fancy word for tissue destruction. And what that does is it creates, we basically can't absorb the water and the nutrients and all this stuff out of the food like we usually should. And that's actually what creates the diarrhea. So this is again in EPEC. EHEC, they're still doing research on it. Not much is understood with regards to one aspect. EPEC is the same as EHEC. It does the tear, it binds the intimin, it does everything. It creates these chains of polymerized actin. The only thing that they don't understand is it does it without phosphorylating. So it does not use tyrosine kinases to phosphorylate. Other than that, it's almost all the same. They just don't understand how it activates it to create the polymerized actin. So that's what research is currently going on EHEC. EPEC, they figured out about, I would say in 1999, that this is what's going on. EHEC, again, a little different. They're still trying the works of figuring out exactly what activates the NWASP. Because it's not a tyrosine kinase, so it's not a phosphorylated tear. So what is it? We don't know. Hopefully they'll have an answer for us very soon. That'll probably help with antibiotics. So that is my interaction that I wanted to discuss. Thank you very much. And my references. I'll be attaching these in the assignment. Thank you so much. A small correction to what I had stated earlier. When I talked about how this all first starts, the very first thing I had said is that the EPEC binds to the intestinal cell through bundle-forming pili. Bind's not the best word. The better word would be it adheres, because it's not considered bound until the intimin binds to the tear. So once the intimin has bound to the tear, then it's considered bind, which is much more tight. It's on there much better. It's on there much looser in the adherence phase. Okay, just small correction. Thank you.